am not that unique. This is an enzyme system which is very fascinating. It's not always that one needs to actually back up with experimental validation. My own contribution to Indian society would be good if I work on problems that are relevant to Indian population. SARS-CoV-2 virus, CRISPR-Cas, Covishield genome editing, circular economy, combination of AI in biological sciences. Computational biology is not only about making predictions. Naturally, all the proteins inside a cells, they tend to unfold.
What is your skincare routine, sir? Do you have a routine?
I think it's an exciting time to be in.
How a person who studied masters in physics, also bachelor's physics, mathematics and chemistry, transitioned to becoming a biologist who earned one of the highest ranked award, Shanti Swarup Bhatnagar award in biology field. Like, how did this transition happen? Like, was it like you were always interested in biology, or something particular thing that sparked the biologist inside you?
So nothing of that kind. So basically, I am not that unique. There are many people like me who have studied other subjects than biology, and then eventually have entered biology out of sheer interest. And the people who have made even much more profound contributions than I have in the field of biology. And there are many examples around the world, also in India, that people who have had basic degree in either physics or mathematics or chemistry, but ended up doing very deep biological research.
Yes.
And I guess that happens partly by accident and partly by interest at various stages in one's life.
So, like, you always had an interest in studying proteins, particularly in biology, or was there anything that also interested you in biology?
So the interest was kindled by the fact that there is a very famous biologist called G.N. Ramachandran,
Okay.
and he was supposed to give a talk in Nagpur when I was doing my BSc. So we went for his talk. He didn't show up, but his talk was read out by someone else. But that field, all of us knew that Ramachandran was a celebrity. And today, I mean, at this stage, we all believe that Ramachandran should have won at least one Nobel Prize, if not two. So and he was already a celebrity when I was a BSc student. And that was fascinating for us to start actually reading a little bit about his work and about proteins and structure function aspects and all. So that was one of the triggers that happened at that time.
Yes. Talking about protein-protein interactions, as you mentioned, that is what you have worked on, X-ray, crystallography, and protein-protein interactions. I've also went through the data, and I got to know that you work particularly on TB proteins, the Mycobacterium tuberculosis proteins. So may I know, like, what was the significance and why choosing that particular to study for, and whatever data, the results that we got, what significance role it plays nowadays also in healthcare or in general maybe?
So when I starting out my career, independent career in India, obviously my passion was to actually work on certain things that affect Indian population. And because it's biology and protein structure, I started relating to diseases that affect large number of Indians. During my postdoctoral work, I had worked on protein from Mycobacterium leprae, which causes leprosy. So obviously the interest to Mycobacteria was closer. And when I started in India, there was no one working on the proteins of Mycobacterium tuberculosis, at least from the structure function point of view. So I decided that at least my own contribution to Indian society would be good if I work on problems that are relevant to Indian population. And I chose Mycobacterium tuberculosis as part of that.
Yes, sir. Actually, because nowadays, if I would want to go with studying in my PhD, if I would want to go with protein study, I know I already have lot of work done like by scientists which I can refer. But at that time, protein was much more like a mystery rather than just a complex structure to study. So it was beautiful that you started that, and now we can see how much data we have that we can read from. So speaking in the same manner, I have read about something called as Chaperonins. So and fun fact I read about it is called as Bodyguards of proteins. I read it somewhere. So can you like elaborate on that? Firstly, also like what are Chaperonins in your term in a very simplified manner, and why they are called so?
So there is a class of proteins which is called as molecular chaperones. And chaperones are the proteins which guard proteins against certain kind of undesirable effects in the cell. For example, when the cell has a heat shock or something, then naturally all the proteins inside the cells they tend to unfold.
Yeah.
And if they tend to unfold, then they would start forming aggregates, which is not good for the health of the cell. And molecular chaperones work at that stage in binding to the proteins and prevent their aggregation, so that the cell can still remain healthy, even under stress conditions. That's how the molecular chaperones were discovered, and therefore they are also called as heat shock proteins. So heat shock proteins and molecular chaperones are essentially kind of synonyms. One class of chaperones is called chaperonins. Chaperonins are the chaperones which form large rings. So GroEL is one such chaperonin which forms a large ring. There are 7 subunits which form one ring.
Okay.
Another 7 subunits form another ring, and the 2 rings are stacked back to back to form a 14-meric assembly. That's a GroEL chaperonin.
And like all the proteins will have this structure.
Not all. GroEL class of proteins have
Only
this.
that particular.
Correct. GroEL is a molecular chaperonin that has, and then it binds to unfolded protein into its cavity and guards them against misfolding and aggregation.
And that is definitely important for cell integrity and all, because if that particular thing is hindered, do you like have any example which disease it leads to? If we are speaking in the scientific terms, what particular hindrance can be seen in human body?
There are quite a few. So chaperonins are essential proteins. That is, one cannot have mutations in chaperonins because if one has mutations, then the cell itself cannot survive. Because cell survival, especially its response to any kind of a stress, depends on this kind of chaperones, molecular chaperones, and therefore most of the molecular chaperones are essential for their function, cellular function. GroEL also is similarly essential, and therefore studying its structure and function and all would be an important thing to do.
Yes, we can say that studying about these structures is very important. Even for students like us, choosing this topic would always be a good option for us. And sir, we know you have served on very great positions at CSIR, NCCS, being a former director, and you have worked in labs during your PhD. So the transition, what do you feel about it? Like being in a boardroom, having all these policies, patenting all these works, or working in the lab? What do you still prefer? Do you miss working in the lab still, or would you want to go back and work?
So I have now superannuated from my service, and I'm firmly back in the lab. Yeah. So I cannot say that I miss the lab because I work now full time in my lab. But as it happens to all the senior people in the country, at this stage of our career, our advice is sought in many different kinds of places. So that keeps me busy in going and advising multiple kinds of academic organizations, government, companies, and so on and so forth. So in that sense, I'm away from the lab for a time period that I'm traveling and advising people on different matters. But otherwise, I work in the lab now.
Sir, your recent studies, around what topics they are, like which topics you're working on recently?
So we are working on an enzyme system called Ribonucleotide Reductase. This is an enzyme system which is very fascinating for multiple reasons. It is an enzyme complex that converts ribonucleotide to deoxyribonucleotide. Now, as you know, deoxyribonucleotides are very important. Yeah. They form the basic unit of DNA. So at any point of time, the cell needs to either repair or make a copy of its own DNA. At that time, for example, in cell replication, a copy of the DNA has to be made. And at that time, a lot of deoxyribonucleotides would be needed by the cell. So this is the enzyme complex that actually makes that thing. So we have been trying to study this particular enzyme complex from Mycobacterium tuberculosis, and we have determined its structure. There's a very complex structure. This enzyme also follows what is called as the free radical pathway, and the structure and function of all the steps involved in the conversion of ribonucleotides to deoxyribonucleotides are very fascinating, and that's what we are studying at this stage.
The work must be very prolonged, like intense, or it takes less time to study this whole thing? Like how
No,
much
so this
does it
project we have been working for last about 6-7 years.
take? Okay.
It is only now coming to fruition, and we are writing multiple papers on our observations.
That's wonderful, sir. When I talk about computational biology and structural biology with anyone who is working in the field, most of the times the doubt I get from them, or even if we are having a conversation, is that whenever they have to present a paper in a conference or anywhere, their work they are presenting, they are cross questioned or asked that where are your wet lab data? And they want to know, like, is the significance of wet lab data if that particular person who is working in only computational biology from the start to end, working with in silico and molecular docking. So is there a lot of significance towards wet lab? Because that is their doubt. Why
No,
are
so
they
computational
asking?
biology is we specialize in making predictions and hypotheses about different biological phenomena. And in that sense, if the predictions we make are desirable to be replicated by experiments behind them. So, if we make predictions and they are either validated by experiments or not, that adds a lot of value. But computational biology is not only about making predictions. Computational biology has a much larger role in understanding biology also. Okay, computational biology has a large role. I mean, as you are aware, last year's Nobel Prize went to computational biology for the protein folding problem. There was no experiment to done for testing their predictions because people knew that what they had predicted was highly accurate. So things like that. So we actually also help understanding biological principles by doing computational analysis. It's not always that one needs to actually back up with experimental validation. But there are also many different ways in which we can develop methodologies to understand biological phenomena using computational or theoretical methods.
So talking about the same, recently, like 5 years ago, we were hit with COVID-19, and the significance of biologist was very important in that period. So how did even this protein-protein interaction and protein folding, what significance role did this particular study played during the time of COVID? Because
So COVID, although I mean my own contribution to COVID work is different, there's essentially on epidemiological scale what I did. But in terms specifically for protein-protein interaction, it was extremely important because the COVID virus, what we call as SARS-CoV-2 virus, the severe acute respiratory syndrome, there is SARS, and coronavirus, that is CoV, and the second kind, there's a 2, has on its surface a protein called spike protein, and the spike protein interacts with the human cellular ACE2, the angiotensin converting enzyme of second kind. So that's a receptor on the human cell surface. And when the spike protein of the virus binds to ACE2 receptor on human cells, it is at that time the SARS-CoV-2 virus can enter the human cells. And therefore protein-protein interactions actually play a very major role in trying to understand how the virus gains entry into human cells. Beyond that, the virus replicates itself, forms hundreds or thousands or millions of copies inside a cell. And every step while the virus is making its copies inside a human cell, there are multiple proteins of the human cell that the virus needs to interact with. So protein-protein interactions play a very major role in determining the outcome of a viral infection such as COVID-19.
yes, and this in turn will help us for the vaccine development and all which was done.
Undoubtedly. So the vaccine was made, or a couple of vaccines were made, which were based completely on the spike protein. For example, in India, what we call as Covishield. Covishield is made with the spike protein and an adjuvant, which is a chimpanzee adenovirus vector.
Okay, it was the combination of that
Correct.
which was used over there.
That's a Covishield.
Yes. Besides all this, we know that you have served multiple roles at different institutes. So, over these many years, what do you think, like, what is the future of academics in India? Like, what futuristic aspect you can see, that how our whole institutes and the students who are working in all these different institutes are coming up with new problems, and they are trying to solve it. They have new quests in front of them from all of the things that are done by the scientist itself. So what do you think, what is the future aspect for India in terms of biotechnology? How much will it grow? What is your perspective on that point?
So the future is always exciting to any person if one keeps pace with the recent developments, future always appears to be very exciting,
Yes.
right? And India is no exception. There are a lot of things are happening around, and especially in the biology, biotechnology, life sciences area, there are many things that are happening around the world, and India is not isolated from them.
Yes.
And therefore, for any young researcher who aspires to become either a scientist in the area of life sciences, or is interested in addressing certain problems related to any kind using biological tools, I think it's an exciting time to be in.
Yes. Any particular fields you feel that the students should be focusing more, like students from our generation who are recently stepping our foots into all these domains? Which domain do you feel we should be focusing more?
I don't think I have any particular recipe. Every individual must look at his or her area of interest. But there are many areas which have emerged in the last few years, last 5 or 10 years. Things like genome editing
Yeah.
using CRISPR-Cas, or things like combination of AI in biological sciences, or things like that, or the environmental remediation using biological systems, or things like circular economy. All of it actually are very exciting, and students may choose any of those what students actually have liking for. And therefore, I don't have any recipe which area people should choose from. But while choosing, look at the future, what areas are emerging, and what you think you can actually make an impact by your own contributions in that particular area.
Definitely, sir. So that is very wonderful advice for all of us, that we need to look ourselves at what skills we have, what interests we have. Then ample amount of options are there in current generation, and we already know about these options, what we are stepping into. So yes, definitely we can learn from that perspective. Sir, we actually have a very fun segment with you right now. It's just a short segment, which is like a tradition of our whole podcast, which will be taken by Ali.
Yeah? Sir, what is the most beautiful protein that you have seen or worked with?
I have worked with a chaperonin called GroES, and I think it's beautiful.
Okay. Sir, what is one myth about biology that you are tired of busting?
So, people say that biology can solve all problems of environment and otherwise, or people can engineer enzymes to degrade, for example, plastics and all. We are far away from that, and that myth one would not like to bust. Although it's possible in the long term, but not in immediate future.
Alright. In your view, what is the most underrated discovery in Indian science from the past decade?
So, if you look at not past decade but last 40-50 years, the discovery of G.N. Ramachandran on the structure of collagen, or the subsequent discovery of Ramachandran prod, has been highly underrated in the world. This discovery ought to have won Nobel Prize, which he did not eventually get.
So speaking of collagen, आपका skin इतना clear है. What is your skincare routine, sir? Do you have a routine?
I have no routine.
Okay. Pune weather then? What is the...
I think it's a combination of multiple different factors, probably. Nutrition, environment, food, weather, everything.
And your genes, proteins as well.
And
Sir-
epigenetics and microbiome, of course.
To add. Sir, how would you explain a protein structure to a small kid?
So, if you look at the building blocks of proteins, these are amino acids, and they are joined back to back to form a long chain. But the long chain doesn't remain a linear string, but it folds to form a globular shape. And that shape is what is actually a protein structure.
Sir, okay, wait. One software tool that you can't live without when you're doing structural and computational biology.
So I use many tools, but the structure viewing that I do is on multiple different softwares with which help me in viewing the protein structures, and I use them all the time.
Sir, which viewing software do you use most?
I use multiple, but let us say Pymol is one of the things that I use frequently.
We made a Pymol film in the first year. Sir, Chai or coffee?
Chai.
Chai? Nice. Sir, do you have a place you go to like after to refresh? कहीं जाना है, चाय पीना है? Do you have like a spot?
So I go to our canteen.
Acha. Nice. And sir, last question is, if you weren't a scientist, what would you be doing right now?
So I am interested in trying to understand protein structures and how proteins fold the way they do. And as a bioinformatician, I am very excited to try to understand the physics and chemistry behind protein folding.
But sir, if not this, because again that is scientist. So if not a scientist, what would you be doing?
If not a scientist, I would be involved probably in science advocacy.
Science advocacy. Okay.
Taking scientific principle to politicians, to bureaucrats, to general public and so on so forth.
But आप science छोड़ोगे नहीं.
नहीं.
Always science is there. Okay, sir. We'll wrap up the rapid fire. I just had a question. Do we know? Still, do we know why the proteins are folding in a particular way?
No, we don't know how and why. We can predict the structure now reasonably well using AlphaFold kind of tools, but how and why it does that, we don't know.
Because in your talk also we saw that when a particular if there is a change in a particular ATGC nucleotide, then after 10 nucleotides again we see a change. And I think using a lot of such data and comparing, we have come up with, integrating machine learning, etc. We've come up with a way to predict the structure, but we're still not there yet where we know why it's folding in a certain.
That's correct.
Okay, sir. Thank you, sir. I enjoyed this session. I hope you did too.
Sir, we have already talked about proteins and your work behind it and all. Yes, I have one more question related to that. Before that, I would want to know who, during your studies, during your work, who was someone kind of like a motivation to you, someone who have you idolized during your work, or like you used to look at that person and feel that yes, I should not leave my work. Who was your motivation?
So it's difficult to point out to a single person. There are multiple people whom always one looks to, and different people motivate one to do different kinds of things, right? So in science, of course, I would say that charity begins at home. So my father was a professor of physics. He was in Pune University, then he was in Nagpur University and things like that.
Okay.
So he was one factor. My own PhD supervisor, Professor Vijayan, was a very, very motivating person. And Professor Vijayan's supervisor during his postdoctoral years was a lady called Dorothy Hodgkin, who is a very legendary scientist, and all over the world her contributions are known. She herself won the Nobel Prize. So she was another very motivating person. Achievements of G.N. Ramachandran working with very less resources in India and producing top quality work was also motivating. My postdoctoral boss Wim Hole and his commitment to work on problems related to third world countries was also very motivating. So I can say that a large group of people who have been motivating.
That's wonderful. And talking about your postdoc journey, which was in Netherlands, if I'm not wrong. So what difference do you feel is studying the study or academia culture overseas and India in comparison? What difference do you see?
I think there is not much different per se, but at the base of it, the kind of bureaucracy that one deals with in India, I feel is rather unreasonable compared to some of the Western countries.
Yeah, and we see the last Nobel Prize that India won was, if I'm not wrong, it was in 2019. Yeah, I guess it was in 2019. So
India's last Nobel Prize came in 1930, C.V. Raman.
sir, 2019.
2019 Nobel Prize went to Venki Ramakrishnan, not 19, 2009.
2009. Yeah.
Venki is Indian origin, but it didn't come to India.
Yeah.
He is a professor in Cambridge.
So what do you feel? Why this gap is there that India, because we have innovations coming up. India is also working in many fields related to healthcare, other than healthcare also. But
So I think we have some kind of false impression about Nobel Prizes. Nobel Prize is a recognition that is given to some outstanding work that is done. but there are many people who have not received Nobel Prizes but have done equally profound work. So what we should look at is the kind of profound impact work has made on the global science than the recognition itself. The recognition to get that needs some many other characteristics, including unfortunately things like lobbying and the place that you belong to and things like that. And there have been many Indians who were of the class that we call as Nobel class. There have been many people. Jayendra Ramachandran was one of them. Shambhunath Dey was another person. So all these people had done some very profound work. They were not recognized by Karolinska as a different matter, but the fact that they made actually very deep contributions to the science is extraordinary.
At least we all who are working in this field, at least we know and we recognize their work, and that is more than enough.
And their work is in textbooks all over the world.
Yes.
People actually, when they read textbook, they read their work.
Yes, definitely. What you said, I can totally agree to that point, that recognition is one side, but what we are working, we should work with passion and all. Definitely, sir. It has been really wonderful talking to you, and we are very happy that we got this chance to talk with you about all this, and especially coming here in your vicinity and talking about all these things. We literally feel inspired, and I really hope who will be watching this will definitely feel inspired by this talk. Thank you so much, sir.
Like, share and subscribe Adaptiv channel.